Hello and welcome to this presentation on gene cloning and cloning vectors. Well, we have learned in the previous class on natural methods of recombination in bacteria, followed by the genetic recombination or artificial recombination, that is DNA recombinant technology. Well, in order to proceed with the genetic recombination or genetic engineering, one of the mainstay of genetic engineering has always been the vector molecules, the carrier molecules, which are very important for transferring foreign DNA from one cell to another cell, one plant cell to another cell, another plant cell, and produce transgenic plants or transgenic animals. Under the title gene cloning, if you look at the term, this clone has been derived from the Greek word called as clone, means a twig. The genetical identical copy of a cell is referred to as the clone. The production of exact copies of a particular gene or a DNA sequence using genetic engineering technique. So the main requirement has always been the carrier molecule, which can uh, transfer a foreign DNA from one cell to another cell without hampering, without affecting its characteristics. The DNA containing the target genes is split into into several fragments using a group of enzymes called as restriction enzymes, and these fragments are then inserted into cloning vectors which transfer the foreign DNA into a suitable host cell, thus producing a chimeric cell, recombinant cell. Now, inside the host cell, the recombinant DNA undergoes replication, and thus a bacterial host cell will give rise to colony of cells containing the clone target genes. This is what we have learned, the principle behind genetic engineering. The basic significance behind carrying out this technique has been the particular gene that has been isolated and its nucleotide sequence can be determined. Control the sequences of DNA can be identified and analyzed. Protein enzyme RNA function can be investigated. Mutations can be identified, example, gene defects related to specific disease, and organisms can be engineered for a specific purpose. Now, before moving on to what are vector molecules and what are different types of vector molecules, let us learn little history behind the various scientists contributing to the field of genetic engineering. We know Gregor Mendel in the middle of 19th century who coined the term gene for the factor controlling the inheritance of biological characteristics of an organism. In 1903, W. Sutton proposed that genes reside on chromosomes, and in 1922, gene mapping by T.H. Morgan, and he analyzed the relative positions of 2000 genes on four chromosomes of the fruit fly, Drosophila melanogaster. In 1944, Avery, McLeod, McCarthy, and in 1952, Hershey and Chase explained the molecular nature of the gene to be deoxyribonucleic acid. In 1952 to 1966, the structure of DNA, genetic code, and the process of transcription and translation was identified. In 1971 to 73, new technology like recombinant DNA technology or genetic engineering or the process of gene cloning was developed. And this technology helped in studying the regulations of genes, aberrations in the genes, and then used genes for production of desired protein. And in 1985, Carey Mullis invented the polymerase chain reaction. Now let us understand what exactly a cloning vector molecule is. It is the central component of gene cloning process wherein a small piece of DNA into which a foreign DNA fragment can be inserted. The insertion of fragment is carried out by treating the vector and the foreign DNA with a restriction enzyme that creates the same overhang than ligating the fragments together. As you can see in the picture, a a picture of a cloning molecule has been shown, which is derived from a plasmid, basically a bacterial derived plasmid. This plasmid, with the help of restriction modification system, it is cut open, and on the both the sides of the open ends you have two overhangs, and basically it is the property of restriction endonucleus that creates normally two types of ends. One is sticky end and another one is blunt end. So depending upon a type of restriction endonuclease, you can produce a sticky end or a blunt end. Sticky ends are desirable. Whereas in case if you are able to produce blunt ends, then you need another set of enzyme which is called as DNA ligase in order to join the two open ends of the DNA molecule. Now, to proceed with the plasmid, this plasmid molecule, once it is cut open with the restriction endonuclease enzyme, then the same restriction endonuclease enzyme is used to cut open to produce a overhang pieces of a foreign DNA which needs to be inserted. Once the overhangs of the foreign DNA are generated, they are ligated with the two open ends of the plasmid vector molecule, and then it is allowed to anneal. Once the process of annealing is over, you can see a chimeric DNA or a recombinant DNA is generated. That's what is the beauty of this genetic engineering process, wherein using a simple vector molecule you are able to transfer any form or any type of foreign DNA from one cell to another cell and try to produce the desirable characteristics in another molecule. Let us learn some of the general characteristics of a cloning vector molecule. A cloning vector is normally, as I said, it could be a fungi derived, virus derived, bacteriophage derived, or a plasmid derived. So, plasmid derived cloning vectors are used immensely in the genetic engineering experiments. Some of the general characteristics, desirable characteristics that a cloning vector molecule should possess are ORI-T sequence. ORI sequence refers to origin of replication and is a specific sequence of nucleotide from where a replication starts. So in general, this is an coding sequence that enables the vector molecule to replicate suitably without affecting any other host cell processing. Then our vector molecule should have a selectable marker gene. Selectable marker gene normally types of antibiotic resistance genes are incorporated into a vector molecule so that a successfully recombinant vector molecule or a chimeric vector molecule can be selected from among the non-chimeric or non-recombinant vector molecule. So in order to do so, a suitable selectable marker gene has to be incorporated. It could be tetracycline resistance gene or chloramphenicol resistance gene or streptomycin resistance gene or ampicillin resistance gene. Then a vector molecule should have restriction sites. Restriction sites basically allows for cleavage of a vector molecule at a suitable place and insert your foreign DNA. Instead, a synthetic multiple cloning site also could be inserted into a vector molecule, which serves as a common site for the action of multiple number of restriction endonucleases. That means multiple cloning site contains multiple restriction sites for number of restriction enzymes which can be cleaved into two fragments and then can be annealed with the foreign DNA. Thus, the successfully created recombinant vector molecule or chimeric vector molecule is transformed into a host cell and then allowed to successfully replicate so that multiple copies of the recombinant DNA molecule are produced. And normally the vector molecule which are used are less than 10 kilobytes in size. Okay, let me show you the diagrammatic representation of general desirable characteristics of our vector molecule that it should possess. You can see origin of replication. This is the origin of replication sequence that allows the replication of the chimeric vector molecule in suitable host cell without interrupting, without affecting the host physiology. Then there are selectable marker genes. As you can see, here lies the ampicillin resistance gene. Here lies the tetracycline resistance gene. Now there is a one fix in this. That fix is that if a restriction site of any of the restriction endonuclease lies within the selectable marker gene region, then it deactivates that selectable marker gene. Thus, the selection of chimeric vector molecule by placing it on a minimal media containing minimum amount of a suitable antibiotic fails. Okay, so in that case, as you can see here, the restriction site lies for Providentia stuarti 1. Then here the restriction site lies for Proteus vulgaris 1. Here there is a restriction site for E. coli R1. Here is the restriction site for Haemophilus influenzae D3 strain of restriction endonuclease. Likewise, Bacillus amyloliquefaciens H1, Staphylococcus aureus 1 here, then Salmonella typhi restriction endonuclease here, etc. So these are various restriction sites which have been shown on the vector molecule. And then as you can see, there are the on the 50% of the surface of this vector molecule is containing the restriction endonuclease sites, thus enabling the vector molecule not to be so useful. In the sense, in case, as I said, restriction sites lies within any gene region, then that gene gets deactivated. Thus, in order to ensure that such of the process doesn't happen, then what we can do is we can have a multiple cloning site in any other region, wherein this multiple cloning site bears region for number of restriction endonuclease. Thus that particular site can be cut open with the help of restriction enzyme, and your foreign DNA can be incorporated successfully without hampering the selectable marker gene region. Coming to the types of cloning vectors, um, where they could be plasmid derived, bacteriophage derived, they could be cosmids, phasmids, and yeast derived vector molecules. Now let us discuss one by one. First type is plasmid vector, plasmid based vector. As you all know, plasmid are basically the self-replicating double-stranded extra chromosomal DNA molecules which are present in a bacterial cell. The advantages of this plasmid based vector molecule would be they are small, easy to handle, easy to purify, straightforward selection strategies, and useful for cloning smaller DNA fragments less than 10 kilobase base pair. Disadvantages less useful for cloning larger DNA fragments as we discuss under the advantages. A plasmid vector for cloning contains an origin of replication as I told you, so that it can allow the replication of an chimeric vector in a host genome. Contains selectable marker genes either between antibiotic resistance or blue-white screening gene, lacks Z gene that allows blue-white screening of the chimeric vector molecules, contains a multiple cloning site, easy to be isolated from the host cell, and plasmids range in size from 1 kilobase pair to 250 kilobase pair. And the examples of some of the successfully extensively used plasmid based vectors has been PUC8, which is 2.1 kilobase pair. Coming on to the various types of plasmid based vector system, the first of its kind of plasmid based vector that was developed in 1977 by Bolivar and Rodriguez, referred to as PBR 322. The P indicates that it is a plasmid based vector, and BR indicates Bolivar and Rodriguez, who are the inventor of or the creator of this very plasmid based vector. 322 distinguishes it from other plasmids produced in the same laboratory, example PBR 325, 327 and 328, and basically it indicates the order in which they were produced, they were created. PBr322 is basically 4363 base pair in size, which is less than 10 kilo base pair. It carries two sets of antibiotic resistance gene, either ampicillin or tetracycline can be used as a selectable marker. Each of the marker gene carries unique restriction sites and insertion DNA into this sec. And thus insertion of your foreign DNA into one of these sites inactivates the specific marker site. It has a high copy number. That means following the successful insertion of a foreign DNA into a vector molecule, nearly about 15 molecules are present in transform cells, but it can be increased to 1000 to 3000 by plasmid amplification in the presence of protein synthesis inhibitor, example chloramphenicol. The vector comprises of DNA derived from three different naturally occurring plasmids. The ampicillin gene is from R1 plasmid, tetracycline gene is from R6-5 plasmid, and the origin replication is from PMB1 plasmid. Let us look at the general features of a PBr322. Have a look at the schematic representation of a typical PBr322, a 4361 base pair plasmid molecule. As you can see, this is a ampicillin resistance gene region, this is tetracycline resistance gene region. Here is the replication or origin of replication. As you can see here, ampicillin resistance gene contains number of restriction sites. Providentia, Stuarta, Proteus vulgare, then ECO311, etc. And then on the other side, you can see tetracycline resistance gene, the ECO322, Bacillus aeroliquefaciens H1, and ECO521, etc. The various restriction sites lies on tetracycline gene region. As I said, if you are using any of these restriction sites, then your foreign DNA will be inserted into this very region that inactivates the tetracycline resistance gene. So in other words, we can say the selection of transformed or a chimeric vector molecule can only be done by placing these chimeric vector molecules on a media containing minimum amount of ampicillin resistance gene, because this vector molecule would have lost the resistance against tetracycline gene. As you can see in this picture, diagrammatically we have depicted how this ampicillin resistance gene and tetracycline resistance gene are going to favor the selection of transformed vector molecule from non-transformed vector molecule. Another vector molecule would be PBr327, which was produced by removing 1089 base pair segment from PBr322. Thus, you can call it as this is a disarmed plasmid, disarmed PBr322. In order to increase the size of the foreign DNA that can be accommodated within the PBr322, a PBr327 was created. The ampicillin and tetracycline gene regions have been kept intact, and in comparison with the PBr322, this has a high copy number, that is 30 to 45 molecules per E. coli cells are amplified. Thus, more the copies of the clone genes, more will be the effect of clone gene on the host cell detectable. The deletion destroys the conjugative ability of the vector, which is important for biological containment. The another set of another set of vector molecule that is derived from PBr322 is PUC vector. Now, as you know, PBr322 requires double screening. PBr322 has limited number of restriction sites, but the newly created PUC vectors for these reasons on the left was engineered. The POC vector lacks selection plasmid. It is a 2750 base pair in size and is one of the most popular E. coli cloning vector derived from PBr322. The nucleotide sequence of ampicillin gene has been changed so that it no longer contains the unique restriction sites. The restriction sites are clustered into LAGZ gene, and it has a high copy number of 500 to 700 molecules per cell even before amplification, and the identification of the recombinant cells can be achieved by a single step process by plating onto an agar medium containing ampicillin and X-gal as the substrate. Thank you very much.